ATTR-CM is a severe and progressive heart condition that can lead to death, increased risk of heart failure and hospitalizations, and decline in quality of life. ATTR-CM is caused by destabilization and misfolding of the TTR, or prealbumin, protein. Pathogenic variants in the TTR gene may lead to increased TTR destabilization, whereas protective variants, such as T119M, have been associated with TTR stabilization. By stabilizing TTR, its dissociation into monomers is minimized or reduced. Studies have found T119M to be correlated with a decreased risk of vascular disease and increased life expectancy in the general population. Attruby was intentionally designed to mimic the TTR stabilizing effects of the T119M variant. By mimicking the properties of the T119M variant, Attruby is able to form hydrogen bonds at the thyroxine binding sites of the TTR tetramer, delivering greater than 90% TTR stabilization as seen in vitro. The enthalpic binding of the hydrogen bonds impacts binding affinity and binding site occupancy, leading to efficient stabilization of the TTR proteins, which can be measured by increases in serum TTR levels. This ultimately reduces the dissociation of the TTR tetramer into monomers, subsequently preventing the buildup of toxic amyloid fibrils in the myocardium. Indication and important safety information. Indication: Attruby, acoramidis, is indicated for the treatment of the cardiomyopathy of wild-type or variant transthyretin-mediated amyloidosis, ATTR-CM, in adults to reduce cardiovascular death and cardiovascular-related hospitalization. Important safety information. Adverse reactions: diarrhea, 11.6% versus 7.6%, and upper abdominal pain, 5.5% versus 1.4%, were reported in patients treated with Attruby versus placebo, respectively. The majority of these adverse reactions were mild and resolved without drug discontinuation. Discontinuation rates due to adverse events were similar between patients treated with Attruby versus placebo, 9.3% and 8.5%, respectively. Laboratory tests. Mean increase in serum creatinine of 0.2 and 0.0 milligrams per deciliter, and a mean decrease in eGFR of 8.2 and 0.7 milliliters per minute per 1.73 square meters was observed in the adults with ATTR-CM treated with Attruby versus placebo, respectively, at day 28 and then stabilized. These changes were reversible after treatment discontinuation. Use in specific populations. Pregnancy and lactation. There are no data on the use of Attruby in pregnant women. Animal data have not shown developmental risk associated with the use of Attruby in pregnancy. There are no available data on the presence of Attruby in either human or animal milk, or the effects of the drug on the breastfed infant or maternal milk production. Please see full prescribing information for Attruby at attruby.com/PI. See the impact Attruby could have on patients with ATTR-CM. Learn more at attrubyhcp.com.